organize the solenoids into a rosette,
Oh,
and that rosette attaches to the scaffolding protein.
oh, okay.
Okay, questions about that so far? Okay, so what's the point of talking about this? Again, the point of this is when we want to start moving around our chromosomes, we have to have this hierarchical structure because we need to make our DNA molecule robust and less fragile. So we just coil it all up in these structures to make a chromosome that looks like this. Okay. I want to define these terms again so that there is no confusion, which means on the exam, like, you can expect questions like, which of the following is defined as division of the cell? Which is defined as division of the nucleus, right? So cytokinesis is cellular division. That's taking a cell and splitting it in two. Mitosis is specifically division of the nucleus, right? Cytokinesis, division of the cell. Mitosis, division of the nucleus. Please remember that when cells enter the cell cycle, the first phase of the cell cycle is interphase, and this is not a phase of mitosis. Interphase is that phase where we're making all the preparations necessary for mitosis and cytokinesis. So that's where our DNA is going to be replicated. That's in the S phase, right? It's where we make all of our, we replicate all of our organelles. That's both in the G1 and the G2 phases. So when you have a chromosome that consists of two sister chromatids, those sister chromatids actually have a physical attachment. And they're attached by a little protein called a cohesin. And cohesins ought to be thought of as like a zipper. That's really what they function as. So when the cohesins are attached to both, sister chromatids, it's like they're zipped together. And that begins occurring in interphase as well.
Wait, so where is that at on there?
The cohesins?
Yeah.
So cohesins would be like a little zipper or stitches here holding these two chromatids together. Okay, so what we're going to do now is go through some phases of mitosis. And we talk about them as if, again, they're discrete phases, but they're really not. Like, there are some specific things that happen throughout the mitotic division, and we've kind of, for the sake of simplicity, organized them into phases, but it's really just one contiguous process. So, on the exam, you can expect questions like, in which of the following phases does this particular event occur, right? So in which phase do chromosomes begin to condense type questions? That would be prophase. What I'd like to do over here on this side of the board is kind of diagram phases of mitosis. And I'm going to show you a cell that has 4 chromosomes. Now, we'll use an animal cell as an example. During prophase, there is a well-defined nucleus . And you have like chromatin, right? And this chromatin is going to begin condensing. And so the first thing to happen is our long chromosomes, they'll look something like this. They're just all kind of loosey-goosey, begin to condense. So that's step number 1. Now, ultimately, we're going to need to separate sister chromatids from each other, and so the nucleus needs to dissolve. We just need to get rid of our nucleus altogether. And so that begins to happen in prophase. this nuclear envelope begins to fall apart like this. You get discrete holes in the nuclear envelope, and that's easier for me to do on the whiteboard, obviously, than it is for you in your notes. But it begins to break apart a little bit. Another really important diagnostic character for prophase is the nucleolus disappears. Does anybody remember what the nucleolus is?
Inside the nucleus.
What was that? I liked it. I mean, it was.
The sac that protects the nucleus.
Sorry, what was that, Matt?
The sac that protects the nucleus.
So the nucleus is kind of a sac. Yeah. And the nucleolus is a region within the nucleus. And what were you saying, Carly?
And it contains the DNA that holds it all together.
So it's a condensed region of DNA that's specifically responsible for making RNA, ribosomal RNA, right? And so that condensed region of DNA disappears. Now, there are some organelles that we talked about called centrioles. And after the G2 phase, each cell has 2 centrioles. You guys remember what centrioles are for? I'll draw the structure. Maybe this will jog your memory. Remember seeing something kind of like that? Had something to do with the cytoskeleton. Remember? So this was the structure from which microtubules grew. So you have 2 nucleosomes, or excuse me, you have 2 centrioles, and this is going to be part of something called the mitotic spindle. So the cytoskeleton that's going to start moving chromosomes around the cell starts growing from the centriole. So we're going to get these.
So are those centrioles?
Yeah, this is a centriole right here. Those green structures are centrioles. And you get little microtubules that begin growing out from the centrioles like this. Now, I'm going to draw microtubules in 2 different colors, but they're just microtubules. And they serve an important function. And it's easier for me to talk about their function with different colors. Okay. So those things happen in prophase, right? Chromosomes begin to condense. The nucleus begins to fall apart. Centrioles, the nucleolus dissolves and disappears. The centrioles begin to migrate to opposite ends of the cell and start growing out microtubules for the mitotic spindle. Now, ultimately what's going to happen is those mitotic spindle microtubules are going to grow out, and they're going to attach to the centriole of each chromosome. And there's a particular protein that facilitates that attachment. It's called a kinetochore protein. And what you're going to get is you're going to get microtubules from each centriole growing to each chromosome. And I'll show you that better in the next diagram. But this is prophase.
I have a question.
Okay.
So the kinetochore protein is that what's made to attach to the chromosomes?
Yeah. So on this diagram of a chromosome over here, what you get on either side of the centriole is a little protein that sits like right here on either side of the centromere, and that's the kinetochore.
Oh, okay.
That's how it's spelled. Nope. Kineto. You bet. It's spelled Kinetochore. And so what's going to happen is the microtubule is going to grow over here and attach to that kinetochore. And it just becomes like a trailer hitch, basically. So think about it that way. Okay, so that's prophase. Questions about prophase?
It all happens at once, right? Like,
Yeah, all this stuff's happening
Okay.
simultaneously. The next phase is called metaphase. So in metaphase, at this point, the nucleus is completely dissolved, and that mitotic spindle has grown out and attached to all of the chromosomes. And those chromosomes begin lining up in the middle of the cell. So that looks something like this. We have our big cell. And our chromosomes at this point are completely condensed. So they're these tight little X-looking things like this. Now, those two different colored microtubules have two different destinies or two different functions. The blue ones actually grow out and attach to the chromosomes like this. And you're going to get each centriole attached to each chromosome. What's interesting though is as soon as one of those microtubules attaches, it starts pulling it towards its centriole. So, for example, I know again this is easier to do on the board than it is in your notes, but just bear with me and watch for just a few minutes. Let's say that this centriole had attached to that chromosome before this one had a chance to. Then what happens is this centriole automatically begins pulling it towards itself. So that it could be that before this one has a chance to attach, we get this one migrating way over here. What that means is this centriole is going to have to grow out an even longer microtubule and attach to it way over there. When that happens, a tug of war begins, and they're both trying to pull them closer to each other, or to themselves, and it always ends up in a draw. So it always ends up so that each chromosome is at the exact center of the cell. So ultimately, that chromosome is going to be pulled back over, the chromosome is going to be pulled back over here, like this. Okay, so those are the blue microtubules. There are also some orange microtubules. Structurally, they're nearly identical. What they do is they provide an anchor. And so they're going to grow out, and they're going to attach to microtubules from the opposing centriole, like this. So that when centrioles begin pulling on those sister chromatids to try and pull them apart, the centrioles don't just get pulled close together. They actually kind of push out against each other with those orange microtubules.
so the orange keeps the centrioles from coming closer.
Exactly right, yeah.
Is there just a meaning for these two types of microtubules?
No.
Or
Just microtubules. Okay, so do you guys know what trisomy 21 actually is? What Down syndrome actually is, like molecularly, what that means?
Like, don't you have a less of a chromosome?
What's
Like
that?
one less chromosome?
You have one extra
Oh,
chromosome.
one extra.
Yeah. The 21st chromosome, you have an additional copy of it. That occurs because of problems at this phase. When microtubules don't grow out and attach to both chromosomes correctly, then you're not going to get separation of those sister chromatids, and one cell is going to end up with an extra copy of the 23rd or 21st or 23rd chromosome, and both types of non-disjunction events occur at the 21st and the 23rd chromosome. Yeah, Carly.
I have a question about that. So if one ends up with more, the other one should end up with one less.
Correct.
Is there a thing that happens with that, or just?
That zygote wouldn't develop into a fetus.
Okay.
Yeah. Okay. So after metaphase, we then get an equally simple phase called anaphase. And with anaphase, what happens is those cohesin proteins come apart that are holding sister chromatids together. It unzips, and the sister chromatids are pulled to opposite ends of the cell, with the centromere kind of leading the way. So that looks kind of like this. Our chromosomes have been pulled apart so that now each just consists of a single sister chromatid. And they're pulled towards the centrioles, with that centromere kind of leading the way and the arm and the leg dragging behind. That looks like this. And those other microtubules are still holding the centrioles in place. Okay, and that's anaphase. The last stage in the cell cycle is called telophase. And telophase is like the exact opposite of prophase. Everything that happened in prophase is undone in telophase. So at this point, we have two sets of chromosomes. And they're no longer X's, right? I mean, they're just kind of individual chromosomes. But they're on opposite ends of the cell, like that.